coming in, it is, but when I look at the numbers for attendance overall, it's about the same. You know, so the people who aren't coming in are usually online or watching the videos. Anyway, we're recording, so I guess the only other reminders to give you will be lab today. Don't forget you need to be there. Physically come to lab. Bring your computer. I got a question earlier. It was a great question. Is it too late to do the lab so you don't have to come in today? You know, officially, I would say yes, it is too late. Officially, because I don't want to make promises I can't keep. Unofficially, if you do it and I grade it and it's good, then I'll send you an email and say, hey, you don't need to come in. But the reason, again, I don't want to make a promise to that is because if you all decide to do it, because most of you didn't do the lab yet, which is okay, you don't have to, but if you all decide to do it like after class today, then there's a large chance, a big chance that I won't be able to get to it before lab. So yes, go ahead and try it, but I won't promise that I can get it graded. What else for lab? That's it for lab. Yeah, and then I guess just another random reminder as I go turn off the light. The exams are out there for you to redo. I know some of you are concerned about your grades. Like you've actually sent me emails saying that you're concerned. Or some of you should be concerned and don't seem to be. But anyway, redo the exams. I don't know, if I was a student, especially if I had anything lower than a B, especially if I had a D or an F, like I would jump all over that. When someone says, hey, you can retake this exam and if you get a better grade, we'll average the two and it'll bump your grade. And if you get a worse grade, we'll keep your higher grade. Nothing to lose. So I highly recommend doing that. And if you need anything, any other advice or ideas on how you can boost your grade, meet me during office hours. Because I know one of you has expressed some concern over getting your grade pulled up. So yeah, meet with me. Because like I've been saying for weeks now, if you meet with me, you'll also get extra points just for showing up for office hours. So any questions about anything? Okay, let's see. The first word for attendance is going to be players. Give me a second, and I'll show you exactly what I mean. So I know sometimes the audio is hard to hear. Morning. Oh, we're in WSU stuff?
Yeah.
We have one, all right. Anyway, like I said, the first word for attendance is players. You can see it right there on the screen. So don't forget to send me those words before 9 a.m. For those of you who are doing the videos and watching the questions, make sure to read the questions carefully. Anyway, let's talk about, let me back you up to what we talked about. This first main bullet point was obviously some background information so you kind of understand or re-understand the molecule of DNA and the molecules of RNA. We also talked about DNA replication, where you learned about how DNA polymerase does its job. Then we got into the second bullet point, which is where we're going. You're learning how we go from DNA to RNA to protein. And remember, that whole process, first is transcription, which is when we go from DNA to RNA. Then it's translation, which is when we go from RNA to protein. And what we finished with yesterday was we finished completely talking about transcription. So to remind you, transcription is going from DNA to RNA. We're still keeping the same language, right? The same nucleic acid language, where these words are made up of letters. For DNA, the letters are A, T, C, and G. For RNA, the letters are A, U, C, and G. So that's where we left off, right? We left off with transcription. We now have, after going through transcription and after processing that RNA, we are now left with a molecule, an mRNA, and that needs to go into the process of translation, and that's what we're going to talk about. Translation is a little bit more complicated than transcription, so your book kind of breaks it down into two parts. First, we're going to talk about the players, so all the different ingredients, if you want to call it that, all the different parts that are necessary for translation to happen, and then once we're very familiar with all the parts, then we're going to talk about the process itself. So before we jump into translation, are there any leftover questions about DNA replication or transcription? Or anything else for that matter?
Okay, so I do have one question. When you're matching up the DNA to the RNA and you're matching up the letters, for some reason it seemed to me like you match up the letters, but it seemed almost like it was flipped around, but that's not correct.
Well, all you need to remember, for DNA anyway, A always goes with T, C always goes with G. And of course, if we're talking about RNA, like in transcription, so if we're going from DNA to RNA, where you normally would put a T that goes with an A, you would put a U instead. And if I have time, what I'll try to do is come up with maybe a worksheet where I ask you to do a bunch of those, like for extra credit, for independent work. Because once you do it a few times, it really is quite simple. Just remember, A always goes with T, C always goes with G, and you should be good.
I'll try and find an example of what let's do.
Okay. Any other questions? Okay. So let's talk about the players, the parts, so to speak. The first important ingredient for translation is the one we just got finished talking about at the end of the last lecture, which is mRNA. And remember, there's going to be different types of mRNA, so it's kind of easy to remember that mRNA is the messenger RNA, and that's the thing that carries the message itself. But I'm about to introduce you to a few different types of RNA, so make sure you know the difference between them. And again, the first one is the mRNA. When we went through transcription, we read the code on the DNA. That is what we made, right? We made mRNA. The RNA polymerase, that enzyme that attached to the DNA, it read the code and it made a string of mRNA. And then, of course, like we said on Monday, then we did some capping, some tailing, some alternative splicing, and then it left the nucleus. And this is where we are now. So what do we need after the mRNA? Well, we need the machine, if you want to call it that translates the mRNA. You can almost think of it like a 3D printer, maybe. So the mRNA is like just a code that you feed into the machine, and it reads it and says, okay, well, first I'm going to attach this thing, then I'm going to attach this thing, I'm going to attach this thing, and we're going to talk about that machine a little bit later. I'm going to give it a name. Right now, I'm just calling it the machine. Obviously, this is going to require some energy. It's going to require some enzymes. But I am going to put an X to that bullet point. I'm not going to ask you about the fact that it requires energy. And also, your book says ATP, but there's also different forms of energy that we don't even get into in this course. But yeah, so in the back of your mind, keep in mind that this obviously is going to require enzymes and it's going to require energy. And then it requires two other components. Actually, I need to fix this slide. The slide is kind of unorganized. So the machine that does the translating, those are called ribosomes. So it's really horribly laid out. I apologize. And then also we're going to need another kind of RNA. I'm going to tell you what it does later. But it's called transfer RNA, so tRNA. But since this is really horribly laid out, let me just rephrase this whole slide in a better way. What are the ingredients? We need mRNA. We need the machine that translates that code, which is called the ribosomes. And we need something called tRNA. So any questions so far? Those are the three main things we need. Of course, as you can imagine, we're going to talk about these in some detail. There's going to be some extra things you need to know about the ribosomes and some extra things you need to know about the tRNA. But for now, you just need to know those are the three main parts to translation. All right, if there's no questions, let's talk about the ribosomes. You can almost think of it, again, I keep saying it's a machine. You could look at it that way. It's literally building a polypeptide. Another way you could look at it is it's an interpreter. If we're sticking with this idea of transcription and translation, then yes, it's the interpreter because it's the thing doing the translating. It's the thing that's converting the 3-letter nucleic acid words, which you already know from last time are called codons, to the amino acid words. So again, that's a good time to remind you what we talked about or Monday, right? We said the words of nucleic acid, they're 3 letters long, and it's made up of those A's and C's and T's and G's, if we're talking about DNA, or A's, U's, C, A, U, C, and G, if we're talking about RNA. So whichever combination of those 3 letters, those 4 letters that you put together in 3-letter words, that says to bring a particular amino acid. So what's the thing that actually brings the amino acids to the machine? That is what transfer RNA is. So I already told you transfer RNA is one of the parts, but now you know what it does. And you need to know what it does. So remember, mRNA is the message, right? It's the messenger RNA that's getting translated. tRNA is the thing that's bringing the amino acids, because really that's all we're doing. I keep saying build a protein, build a polypeptide. As far as we're concerned in this conversation, that means the same thing. But another way I could say this is we are making a string of amino acids, right? Because that's what a polypeptide is. Or a chain of amino acids, whatever you want to call it. The point here is, this is what we're building. We take one amino acid, and we attach another one to it. And we attach another one to it. That's it. That's all we're doing is attaching a bunch of amino acids one at a time that just link up in a chain. The thing that brings them is called the tRNA. That is what it's transferring. It's transferring the amino acids. The tRNA carries out two functions. Obviously, to do what it needs to do, it should be common sense in a way. First of all, it has to pick up the appropriate amino acid, and then it has to recognize the appropriate code on the mRNA. So it's not like, let me put it this way, it's not like it's a one-size-fits-all. So there's not just a transfer RNA, a tRNA that's doing all of it. But they're very specific. So there's a specific tRNA for each of the 20 amino acids. Right? So it says, all right, I'm the one that carries arginine. There's a tRNA that carries arginine. It has to be able to match that amino acid, and also it has to recognize its code on the mRNA to say, okay, it's time to bring in the arginine. And this will make a lot more sense when I show you a picture of it. As far as test questions go, I'm going to put it next to this. That's not going to be specifically a question, but you still need to understand it to understand how tRNA operates, right? How is it transferring these amino acids to the ribosomes? And your book says that it's the unique structure that enables them to do both. So again, here we go back to the structure and function thing that we've been talking about all semester. The next slide, it's going to make a little bit more sense. Just kind of show you a picture of this a little bit. But are there any questions about this slide before we move forward? All right. I will underline the next word for attention. Attention, good grief. The next word for attendance. I'm going to underline it right here. Put a little arrow to it. That's the next word for attendance. You know, sometimes I don't like to say it. So there it is, the next word for attendance. Anyway, we're still talking about the tRNA. Some things you need to, well, I'll say you need to know this. It's made up of a single RNA strand. Not that that matters too much to you guys. Like, what's the matter to you if it's made up of one RNA strand or two RNA strands or even 100? It doesn't matter, but just so you know, it is made up of a single RNA strand. I'll put it next to that just so you know, because I'm not going to test you on that. It does twist and fold upon itself. So the reason that's even worth mentioning, the second bullet point, is because when you look at this picture, this looks like a double strand of RNA. This looks like a double strand of RNA. This looks like a double strand of RNA. You can see they're hydrogen bonded to themselves, just like a double strand of DNA. However, technically, again, this is not a double strand of RNA. This is just a big, long, single strand that's kind of folded up upon itself. And I'm going to put it next to that too, because again, I'm not going to test you on this. It's just if you're really, truly going to understand how it functions, you need to understand its structure. And that's why we're talking about this. I should also point out that this picture is not from your textbook. So if you're seeing some stuff and you're like, what is the D loop? What is the anticodon loop? Actually, we'll talk about that. And what are these symbols, right? Don't worry about those. You can look them up for independent work if you want, but that goes a lot deeper than even the biology majors get at a 100 level course. Anyway, when it folds up on itself and it twists and it double bonds, or excuse me, hydrogen bonds to itself, then it forms double-stranded regions, like I pointed out here, where short stretches of RNA pair with other stretches. So again, as far as the exam is concerned, I just put a big old X through this whole slide, meaning there's not going to be specific questions about any of this information. But again, to truly understand how the tRNA works, you need to understand how it's shaped. So are there any questions about this slide? All right, let's talk about it a little bit more. At one end of this tRNA is a triplet of bases called the anticodon, and that is complementary to the codon on the mRNA. I'm gonna put a big question mark on this. I doubt I'll get into that much detail, like, what are all the different parts of the tRNA? But you definitely need to know what an anticodon is, just so when I use it in conversation or when you read about it, you understand what I'm talking about. So that is the thing on the tRNA. Because remember, let me back up. I said the tRNA needs to do two main things. It needs to pick up the appropriate amino acid, right? Remember, there's 20 of them, and it's very specific to which one it picks up. And it has to recognize its code on the mRNA. So if it's the one that carries around tryptophan, for example, it needs to recognize, like it's carrying it around. On the other end of it, on the anticodon loop, it needs to say, oh, that's my code. That's the thing that says I need to bring this tryptophan in. And that's what the anticodon is. Anyway, so on one end, again, you have the thing that recognizes, hey, that's me. On the other end, that's the end that actually carries the specific amino acid. And now that I'm thinking about this, I really don't like this PowerPoint. I'm going to make all kinds of changes. These things can be swapped, in my opinion. Of course, I teach from the book, and the book probably presented it in that order. But I think it'd make more sense if I told you on one end, it's carrying its specific amino acid. On the other end, it has the anticodon that says, all right, it's time for me to bring in my specific amino acid. And this is going to make a lot more sense when I show you this next slide. Then after that, I'll ask if you have any questions about this. But here's what it really looks like, right? Here's a more simplified version of what it looks like, because this is a little bit more easier to understand, and that kind of makes sense. So again, on one end, here's the thing that says, all right, I need this specific amino acid, right? There's 20 different amino acids, so this, even though it just looks like a purple line, technically that would be different, right? There'd be 20 different versions of this, so it can carry the specific amino acid. And then again, depending on which specific amino acid it has a particular codon or anticodon to recognize, all right, this is me, I need to bring in this amino acid. So again, the mRNA has the codon, and the codon would say, hey, give me this amino acid. And one particular tRNA would say, oh, that's me. And would come and say, here you go, here's amino acid. And then it would shift over, and we'd look at the next codon and say, bring me that amino acid. And a different tRNA would say, oh, that's me, I'm the one carrying that. And it would come, and it would bring that particular amino acid. It literally is just the code saying, bring in the amino acid, and then the specific tRNA brings in the amino acid. Does that make sense to everybody? And of course, I just got into describing the process a little bit. And technically, we haven't even gotten there yet. So technically, we're still talking about the players, not the process. But let's talk about the next part, the ribosomes. First of all, ribosomes are technically organelles. I'm not going to test you on that. But yes, they are technically organelles. You look that up for independent work. Like, what's the difference then? Why are those things organelles and the other things aren't? Good independent work topic, but we don't have time to talk about it. In a sense, this is the thing that's coordinating the functions of the mRNA and the tRNA. Because like I said, the mRNA says, hey, I need this amino acid, and then the tRNA says, oh, that's me, I've got that one, and it comes in, right? And then we get the next 3 letters on the mRNA that says, hey, bring that amino acid, and then a particular tRNA says, oh yeah, that's me, that's my amino acid, and it comes, right? So I already told you that. And now what I'm telling you is it's the ribosomes that coordinate these things. It's not like the mRNA is just out there floating around in your cell, and then the tRNA just come in and do it. It'd be a lot simpler, but that's not how it works, right? It's the ribosomes that do that. So using my silly little example, technically, it's the ribosomes holding the mRNA saying, okay, oh, we need this amino acid. That's what the ribosome says, right? The mRNA doesn't say it. The ribosome reads the mRNA and says, we need this amino acid. And the tRNA says, oh, that's me. And it comes in and it brings it in. That's how that works. More importantly, I guess if anything, you might say the most important thing to remember about the ribosomes is this is the thing that's actually making the polypeptides. Right, so we're bringing in amino acid after amino acid after amino acid. Right? We're just adding these amino acids to each other, a big long chain of amino acids. That's the thing that's actually making the polypeptides. Technically, each ribosome is made up of 2 subunits. Put an X to that, so I'm not going to test you on that. This will make sense though, because you can see, once I show you how it works, you can see how you need 2 parts coming together. But yeah, technically it's 2 parts. It's made up of proteins and then yet another type of RNA called ribosomal RNA. And we'll underline that, because even though I'm not technically going to ask you, you don't technically need to understand that it's made of 2 parts. You do need to understand the 3rd type of RNA. So we have mRNA that we talked about first, which is the message. It's the thing with the codes. We have the tRNA that transfers in those amino acids when called upon to do so. And then finally, the 3rd And final for us, type of RNA we're going to talk about is the rRNA, the ribosomal RNA. So if you take notes, just keep that in mind. We've now covered all three types of RNA that will be on the exam: mRNA, tRNA, rRNA. And for independent work, you can look this up. There's also a bunch of other types of RNA, stuff we don't get into, siRNA, all kinds of stuff. It's actually pretty interesting in my opinion. Sometimes RNA acts as enzymes, and sometimes, well, we don't have time to get into it. It's really interesting. So these ribosomes, obviously, if they're going to coordinate the functioning of the mRNA and the tRNA, obviously they need binding sites. So they need binding sites for the mRNA, and they need binding sites for the tRNA, because again, they're just bringing it together. The ribosome says, all right, it's reading the mRNA. It says, all right, we need this amino acid. So it needs a place for the mRNA to come in, and it needs a place for that tRNA to come in and bring the amino acid. And again, the fact that it has two parts, two subunits, doesn't matter to me. So I'm going to put it next to that. The common sense should just tell you if this thing is coordinating the function of mRNA and tRNA, then it has to have a place for each of those to bind. And yes, technically the mRNA goes on the small part, tRNA goes on the large part. But this is a 100 level class for non-biology majors, so I'm not going to get into that much detail. Plus, this is the picture that your book gives you, because we, again, are giving you the simplified version. This is actually not accurate. So if you were to look this up, there's actually a whole different site that we don't even talk about in this course. So again, you're getting the Cliff Notes version. You're getting enough information to understand the basics of how it works. So, if you ever had to deal with looking up how a virus works or how a particular vaccine works, you'll have enough ground level information to do some research without having to start from scratch. But this is not 100% accurate. It's just a simplified version. Anyway, here's everything we said, right? This is the RNA, this is the ribosome, right? This is the thing that builds the polypeptide. This is the thing that coordinates the mRNA and the tRNA, right? This is the thing that has a large subunit and a small subunit. You can also see that there's a P site and an A site. And now I'm going to talk about what happens in the P site and the A site. But I'm not going to specifically ask you anything about the P site or the A site. That'll make sense later when I talk about the process of translation. But, yeah, that's just where the tRNA is lying. Anyway, any questions about these parts? All the parts are in this picture now, right? The RNA, the tRNA, the mRNA, the ribosomes, it's all there. That's those are all the players. No questions? Okay. Let's talk about the process now that we know about the players. Process of translation. Here's some good news. Does anybody remember the 3 stages of transcription, which is what we talked about on Monday? Remember I told you there's 3 stages. You need to know what they are in order, and you need to know what happens in each 3. Does anybody remember the name of the 3 stages of transcription? Anybody online? That's all right. I only told you once, and I don't expect you to be studying yet. Wouldn't hurt, but I don't expect it. Anyway, the same 3 stages for transcription are also the same for translation, right? Initiation, elongation, and termination. They are slightly different in the way they operate. Good news for you biology students, anyway, is that they are called the same thing. So again, understand, or remember, initiation, elongation, termination. And that's what we're about to talk about, the processes of initiation, elongation, and termination. And this should make sense. If we had more time, I would sit and literally just wait for somebody to come up with these answers. But we'll see. But think about this. Initiation of transcription is when we have the DNA, right? And the RNA polymerase attaches to it, because the RNA polymerase is the thing that reads the code and makes the RNA. So that's what happens in initiation of transcription. The RNA polymerase attaches to the DNA, right? So any guesses what might happen now that you know the parts, right? You know the players of translation. Does anybody want to guess what might happen in initiation of translation? Even though I'm not taught you yet, but you do know the parts, and you do know what happens in the initiation of transcription. And again, if we had more time, I would literally just sit and wait. Because surely you guys can figure this out. It's really not that complicated. So just the same way initiation of transcription brings the parts together, so does initiation of translation. The parts are different, right? So the two parts of transcription are the DNA and then the RNA polymerase that attaches to it. Here it's the same thing, we're just looking at different parts. So it's bringing together the mRNA, which obviously we talked about earlier, have the cap and tail to help it do that job. And then of course we have the ribosome and the amino acids. So all the parts are coming together. So in a sense, when it comes to studying, this part's pretty simple, right? It's the same, essentially it's the same as transcription of, or initiation of transcription. It's bringing parts together. So any questions on initiation? And to remind you, when I talk about the mRNA, this is what it looks like. We've already seen this because at the end of our discussion of transcription, we said it has to be processed. So remember, we had the mRNA strand, we added a cap, we added a tail, we did some splicing, and this is what we ended up with. There it is again. Your book gets pretty specific now. So like I said, initiation is all the parts coming together, but your book gets very specific. Oh, sorry, here, look, this picture is not from your textbook. This picture is from a different textbook, and you can see here that we have an extra site. So you don't notice, no need to study this picture, especially this one because it's not from your textbook. But let me just say this. This is all giving you a bunch of information that I don't necessarily want you to know, except that, and I'll talk about that in a second. So yes, technically what happens is the mRNA first attaches to the small subunit. And then after that, technically what happens is the first tRNA attaches to the mRNA, right? And then what happens is the large subunit comes in. So the last slide I showed you was more important than this slide as far as how my exam is going to be. All I care about for initiation is that you know all the parts are coming together. Then your book gets specific and tells you what order they come together. And I just put a big X on through there to indicate, don't need to know in what order they come together for the exam. Maybe I'll have that as an extra credit question, but I'm not going to ask you that for, regular season questions, so to speak. But I will say this, where does this all happen? Where does the tRNA, that first tRNA go? It starts at the start codon. That part you do need to know. So if we were comparing and contrasting transcription and translation, right? In transcription, you're bringing the parts together. In translation, you're bringing the parts together. Transcription, it all starts at the promoter, right? The thing we talked about yesterday, or Monday. Remember, that starts at the promoter. Translation, it starts at the start codon. Which makes it, in my opinion, a little bit more difficult to remember and a little bit more complicated. Because if you only had to memorize one or the other, that doesn't seem that hard, right? It all starts at the promoter, or it all starts at the codon. That's kind of easy. Now, for the exam, you've got to differentiate between the two. Because I can guarantee you when I ask the question about initiation for transcription, correct answer will be promoter, but one of the wrong choices will be start codon. And it'll be an easy mistake to make, and I apologize. I wouldn't call it a trick question, but it's as close to a trick question as I get. Because I do realize it's tricky. I do realize that's an easy mistake to make. And as usual, gotta be one of those questions where it's worth more points. So if you were to guess promoter instead of start codon, you probably get half credit. So at least you're on the right track. Then I'm also going to have some words that make no sense. I'm either going to completely make them up, or I'm going to have some words that we've already talked about, like things from photosynthesis and mitosis. So please don't choose those. Anyway, initiation, again, brings all the parts together. Where does it start? It starts at the start codon. For the exam, that's what you need to know. Are there any questions about that, about initiation or translation? The next slide I'm going to show you is this picture at the bottom, except it's from your textbook, so it'll make a little bit more sense. It's not going to have the 3 sites, and the 3' and the 5', and the GTP instead of ATP, all that stuff that you don't know about. So here's a more simplified version. There we go. Again, not that you know it in this order. How does this happen? Well, first of all, the mRNA comes together with a small subunit. The tRNA recognizes that start codon and comes in. Also, side note, I'm gonna put it next to it, because you don't need to know it. But the first amino acid is always that one. Not that it matters. And then once all those parts are together, then the large subunit comes in. And technically it comes in so that that first tRNA is in the P site and the A site is left open. But again, like I said earlier, I'm not gonna ask you about the P site and the A site. So again, for the exam, what do you need to know about initiation? All the parts came together. And it all starts at the start codon. Any questions? Okay, if you download this PowerPoint, you can watch this little animation. Right there, it's a decent one. Hopefully I can find some better ones to share with you. All right, now we're talking about step 2. We're no longer talking about initiation, we're talking about step 2, which is elongation. And I'll give you the next word for attendance, which is going to be first. So if you want to say that in a confusing way, the third word for attendance is first. But anyway, let's talk about it. We're talking about elongation. And just to remind you, because there should be a little bit of comparing and contrasting, when we talked about transcription, elongation was very simple, right? We're elongating the strand of mRNA, because that's the whole purpose of transcription, was to build a strand of mRNA. Likewise, for elongation, what are we trying to build? We're trying to build a string of polypeptides. So for elongation, basically, that's what you need to know, is we're just making the polypeptide longer. We are building amino acids, one at a time. That's all this is, just a string of amino acids attached to each other. That's the good news. The bad news is your book gets into even more detail. So this is stage 2 of translation. But stage 2 itself is broken down into 3 steps. That's the bad news, is that is further broken down into 3 steps. Some good news is I'm not going to ask you to know the 3 steps. You just need to know what happens all together. So sort of like the last thing we talked about with initiation. Remember, I was like, technically, first the mRNA attaches to the small subunit, then the tRNA comes in, then the large subunit comes in, right? That happens in a very specific order, but I told you you don't need to know it that order. Same thing here. I'm going to teach you how this happens in order, but you don't necessarily need to know how it happens in this order. Of course, some of it should be kind of intuitive if you really sat down and thought about it. Hopefully you could come up with this on your own. So step 1 is the codon recognition. In other words, got that tRNA, or let me say it a different way. I'll say it the same way I said it earlier. Here you have this ribosome, and it's basically just reading the code, right? And it reads the codon and says, okay, I need, all right, this doesn't say which amino acid it is, but let's say it's that one. I need a diamond amino acid, right? It reads it and it knows. I need a diamond amino acid. And then the tRNA, the particular one that has the diamond amino acid, not the other ones, it says, oh, that's me. And it comes in because that anticodon on that tRNA matches the codon there, so we know that those things go together. So that's step 1, right? The tRNA comes in, which should make sense. And again, if we had time, I'll just sit and wait for you guys to come up with this. Because logically, if we're adding a bunch of amino acids together for elongation, the first step logically should be, we'll bring in an amino acid, right? If we're adding amino acids together, step 1 should be bring in an amino acid. And I'm going to see if anybody can guess too. I'm going to give you a hint by looking at step 1. So let's say elongation has been going on for a while, right? And it happens. Step 1, step 2, step 3 happens, and it keeps repeating for a while for elongation. So you can see here, it's been going on for a while because we already have a chain of amino acids. We clearly didn't just start because there's more than just one amino acid, right? This has been going on for a while. So we bring in this amino acid. It's right here next to this chain that we already have. What do you think the next step might be? Well, we're bringing in the amino acid. That's step 1. Assume it's already right here. Boom, it made it. That's the end of step 1. That amino acid and that tRNA are just sitting right there. What do you think the next step would be? If we had more time, I would wait, but logically, I'm sure at least some of you have come up with this. because it has to be the next step. Now that new amino acid is right next to the chain, they combine, right? Because we're just adding amino acids. So step 1, bring in the new amino acid. Step 2, attach the new amino acid. It's that simple, right? Step 2 is the peptide bond formation. And again, I'm not going to ask you this. So when you're studying this, it would be helpful to read through it. But definitely don't focus on the name. Like, what is step 1 called? What is step 2 called? Just understand what happens. Because again, for the exam, you need to know what happens. Just not necessarily in this order. Though logically, it has to happen in this order. So that's what happens, right? This one was already in for step 1. So now those covalent bonds are formed. Also a new word for you. Technically, when we're talking about covalent bonds between amino acids, those are called peptide bonds. And I'm going to put it next to that, meaning I'm not going to require you to differentiate. Peptide bond is just a specific type of covalent bond, but I'm not going to ask any questions about that. I just want you to know, if I say peptide bond, just keep in mind that's what I mean. Or if I say covalent bond, know that I'm still correct. I'm just not using the proper terminology, which is peptide bond. The more proper, the more correct. So anyway, again, step 1, which we talked about earlier, bring in the new amino acid. Step 2, attach the new amino acid. Then obviously we need to do some things to get rid of step 3. I wouldn't even ask you this because I don't know, this one's not so logical. But that leaves the tRNA in the P site. So again, I'm not going to ask you this detail, but technically what's happening is you're not attaching the new amino acid to the growing chain. Instead, you can see with this little arrow, the growing chain is being attached to the new amino acid. Technically, that's what's happening. But again, that's a level of detail I don't expect you to know for 100 level non-biology majors. The point here is this main bullet point in bold. For the exam, that's what you need to know. The chain has one more amino acid for step 2. Brought in a new amino acid in step 1, and in step 2, it was attached. But again, that might be some great test extra credit questions. Who knows? I might ask you, was the amino acid added to the chain? Or is the chain added to the amino acid? And the answer is the chain is being added to the amino acid. All right, so any questions about step 2? All right, now picture this. Here we have the little arrow that shows that this chain gets attached to that amino acid, right? So now in your mind, if you can do it, if you can visualize this, imagine this tRNA is just sitting there blank now because nothing's attached to it. And then this tRNA in the A site has a long strand of mRNA. Could create a long strand of amino acids. So this one's a little bit less intuitive, but not still slightly intuitive. Does anybody guess? Can anybody guess what the next step must be? We have this tRNA that is now empty. We have this one that has the whole strand on it. And here's a hint. Let me back up. That's what it looked like to begin with. So any guesses what might happen next? Again, it's hard to visualize because it's not drawn, but this tRNA is now empty. It doesn't have anything attached to it anymore because everything is shifted that way.
Detach?
Yes, good. Because now we don't need that thing anymore. It's done doing its job. Also, we need the A site because as we learned from step 1, the A site is where the new tRNA comes in. So step 3 is translocation. Get rid of that tRNA because it's done. It already gave up, it already did its job. And now we've got this tRNA that has the strand, right, the ever-growing strand of amino acids. And they can just scooch over, right? So then that thing is left open and the whole process can begin again. So again, not that you need to know it in this order, but what happens in elongation is a tRNA brings in a new amino acid. The chain of polypeptides is added to that amino acid, and then things get shifted around so we can start again with step 1. That's basically it. And again, you don't even need to know in that order. If I were studying, or if I were you and I was studying, First thing you should study and memorize before you even talk about all those details is the fact that for a while now, we've only talked about elongation. The most important thing you need to know about elongation is, in elongation, we are adding amino acids, right? Polypeptide is elongated. The strand of amino acids, which is what a polypeptide is elongated. It's getting longer and longer. And then, of course, here's a picture of all three put together. And again, logically, this should make sense, right? We're just adding amino acids. So logically, you need to bring in an amino acid, right? And then it needs to be attached to the strand. And then the slightly less intuitive thing is things need to get shifted, right? The tRNA that's done needs to get out of there. This thing needs to move over so there's room for a new one to come in, and the process continues. And that's it for step 2, which is elongation. Are there any questions about elongation? All right, the next one is a lot easier. Step 3, just like it is in transcription, is called termination. And remember, in transcription, we said the end was called the terminator, right? So when the RNA polymerase is traveling along the DNA, and then it hits that certain code on the DNA that we call the terminator, it said, oh, job's done, and then everything detached and everything went its own way, right? The DNA came back together, the RNA polymerase went away, the mRNA that we just built went on to this process, right? And so in a way, this is very similar. Because in a way, it's the same thing. In that, in termination, there's a certain code that's read, that's reached, and then all the machinery, all the parts know, okay, this process is done, and everything goes its own way, right? The large subunit, the small subunit detach, the tRNAs get released, the new polypeptide gets released, everything goes its own way. Also, by the way, for your knowledge, again, this picture is not from your textbook, so it looks slightly different than what you're used to, that's why. But yeah, so again, for termination, for the exam, all you need to know is that's what everything comes apart. Your book gets a little bit more specific. I'm gonna put it next to this. This might be maybe an extra credit question, but probably not. But anyway, there's 3 different codes. If the code says AUU, UAG, or UGA, that means stop, right? That happens to be the code. Of course, you don't need to know that, but your book talks about it, so do I. Obviously, it doesn't code for an amino acid because it just says stop the process. Yeah, that's it. So again, transcription stops at the terminator. Translation stops at the stop codon. Just like transcription starts at the promoter, translation starts at the start codon. So when we're talking about translation, it's all about the codon. Start codon, stop codon. Very easy to remember. Transcription is a little bit trickier, promoter and terminator. Yeah, that's it for termination. Any questions about that? Okay. I like that your book included this. It's a quick review. So now that you know everything in detail, kind of take a step back, review everything we've learned. So again, the flow of information in this whole process moves from DNA to RNA to protein. And again, when you're studying, I recommend studying that first, memorizing that first. That'll be a test question. And if you don't know that that's the order in which it goes, then who cares about all this other stuff? Who cares about start codons and stop codons and translocation? What's that matter? If you don't remember that the process starts with DNA and then goes to the RNA and then goes to protein, then who cares about that other stuff? That's a very important thing to know, especially in context of the COVID-19 vaccine that uses mRNA. So for people to think that this is gene therapy and that you're getting injected with mRNA, somehow that's going to affect your DNA, that makes no sense. That's why, again, it's a very important thing to know. Know that before you know the details. Anyway, once you have that memorized, then maybe memorize some nomenclature, which is that when we go from DNA to RNA, that is called transcription. Okay? Going from DNA to RNA is called transcription. That happens in the nucleus, at least for eukaryotic cells, right? Prokaryotic cells don't have nuclei, so obviously it doesn't happen that way for prokaryotes, but don't worry about that. And then, of course, you remember that the RNA is processed before it leaves the nucleus. because we have to make sure it doesn't get attacked when it's in the nucleus, right, or when it leaves the nucleus. And we need to make sure that the ribosome recognizes it and says, oh yeah, that's the thing I need to work with. So anyway, then it leaves the nucleus, and then like we already said up here, we go from RNA to protein. What is that called? That's called translation. And that's when one ribosome makes an average size polypeptide. And then your book gets a little bit specific. I think it's the first time I'm telling you this, but it happens in less than a minute for an average size polypeptide. I will put it next to that. I'm not going to ask you that much detail. But again, what you really need to know is translation is when we go from RNA to protein, and it's the ribosome that's doing all that work. And then as we mentioned earlier, a few times earlier, technically a polypeptide is not a protein yet. Technically a polypeptide is just a long chain of amino acids. It's not considered a protein until it folds and assumes its final three-dimensional shape. If you remember back in chapter 3 when we first started talking about this, I told you that, right? I think the example I used was a paper airplane. When it was all folded up, I said, what is this? I said, it's a paper airplane, which you could also say is a piece of paper, right? When it's folded into that shape, it is a piece of paper, and it's also a paper airplane. However, when it's just a piece of paper, it's just a piece of paper, right? It's not an airplane until you fold it. Same thing with polypeptide, right? Polypeptide is just a string of amino acids. It's not considered a protein until you fold it into that proper shape. And even then, it's still considered, it still is a polypeptide, but it's considered a protein because you folded it into that shape. Here's a visualization of everything we said. There's transcription happening in the nucleus. The RNA polymerase attaches to the DNA. It opens it up so it can read the code. It makes a strand of mRNA with the introns and exons and everything. And we add the cap, we add the tail. We do some splicing to get rid of the introns, so we are only left with the exons, which are the parts that are expressed. And it leaves the nucleus. Here we have the parts, right? The tRNA attaches to its particular amino acid. And again, like I said, there was one bullet point that I put it next to, but I said this is going to require enzyme and energy. And again, I told you I'm not going to test you on that. But yes, it takes energy to take this tRNA and attach it to its proper amino acid, but that's more what we're focused on. We have that enzyme, we have that tRNA with this particular amino acid. That then is added with the mRNA and the ribosome. All those parts come together. The code on the mRNA is read. The strand gets longer and longer through elongation. Finally, we hit the stop codon and termination. Everything's over. All the parts go their own way. Then we're done with translation. Actually, we're done with the whole process, right? We went from transcription here to translation there. And I'm going kind of quickly, I realize, because it's just a review. We've literally just got finished talking about this. But then your book points out that transcription and translation are how genes control structures and activities of cells. So it's not that this is going to be a test question. So I'll put it next to it, signifying that. This is why this information is important. So as boring as it may seem to some of you, or maybe most of you, because none of you are biology majors, this is really important, right? This is how we live. Without all this stuff, we wouldn't be alive. So all those other processes we talked about already, mitosis, meiosis, photosynthesis, respiration, all that stuff is controlled by enzymes and different proteins, right? And this is how those proteins are made. So all that other stuff we've learned about wouldn't even happen without this. Of course, you can make it the same amount when you get that deep. Anyway, again, this is just review, put it next to this. The information originates with a specific sequence of nucleotides in the DNA gene, right? That's the instructions to say this is how you build a protein. That gene dictates the transcription of the complementary sequence of nucleotides in the mRNA, which we just said. That mRNA sequence specifies which amino acids are going to be attached, right? It says, all right, bring me this amino acid, bring me this amino acid, right? That's what it says, which we just got finished talking about. And then finally, the proteins from the polypeptide determine the appearance and capabilities of the cell and the organism. So it's a good review, but if you're studying the PowerPoints instead of the book, you just skip that PowerPoint. If there's anything that's on this PowerPoint that you need to know, we've already mentioned earlier. So, any questions about that? We're still doing the review, excuse me. So the DNA to RNA to the protein pathway used to be believed to be the only way genetic information controls traits. However, I'm glad your book points this out, we've recently discovered that there's also more complex roles for RNA. So as I was saying earlier, it's not just mRNA and tRNA and rRNA. Of course, those are the only ones you need to know. But there are some more versions of RNA, and they have very complex roles. And you can read all about that in chapter 11 if you want. But we're going to skip chapter 11. We go from chapter 10 now. I think we're to chapter 13 next. Yeah, so after this, we're skipping 11 and 12 and going straight to evolution. But it's a good time to remind you, the next exam is going to be on chapters 8, 9, and 10. So we're almost done with this, and we're about to have our third exam. It's a good time for me to repeat myself too. If you haven't retaken the first two exams, might as well, right? It's not going to hurt. Anyway, let's talk about when things go wrong. And this is also an important thing to understand about how viruses work. I've also heard a stupid claim that people who get the vaccine are the reasons that we get, whether we're talking about flu or COVID or whatever, they're the reasons we get mutations. And that's not true. I'll explain why after I explain how mutations work on Friday. Then I'll take a step back and we can talk about that from a public health point of view. But the last word for attendance is going to be, let's just say red, to remind you that Friday is a red Friday. If you wear red, on Friday you'll get extra credit, especially if you come and take a picture with the skeletons and the skinless body for Halloween. So any questions? All right. So
It was only 4 attendance words, right?
I honestly didn't keep track. I don't know. What I do is just to make sure I remember correctly is before I, you notice probably, before I email you guys back and say full credit or partial credit or whatever, I always make the questions for the video first. So I go through the video and then I know how many words and what they are. So sorry. Any other questions? That was a good one.